Hello and welcome to Nikolai's genetics lessons, and today we are going to answer 3 simple questions. Use the below structures to answer the questions. And here are the 3 questions. Here we have tripeptide, here is the first amino acid, and this is glutamic acid. If this hydrogen would be lost, this would be glutamate. We call this glutamate in this case. And here is the second amino acid, and here we see silyl group, so this is cysteine. These 2 amino acids can make disulfide bridges, and here's the third amino acid. This is most simple amino acid, glycine. At this alpha carbon, we see it is attached to 2 hydrogens, so this is the most simplest amino acid. Now let's check the first question. Which letter arrow points to the end of the peptide that is the amide end? This end we call amide because we have nitrogen here, so this is going to be answer A for the first question. And second question, which letter arrow points to the end of the peptide that is a carboxyl end? Each individual amino acid has amino end and carboxylic acid end, but when they would join in the polypeptide chain, in our case tripeptide, such single molecule would have amino end and carboxylic end. So the correct answer is answer C to the second question. And question C, which letter arrow points to an amide or peptide bond? Here is the amino acid number 2, here's amino acid number 3, and between them, between this carbon and this nitrogen, we have a peptide bond. And on some pictures in the textbooks, you can find that peptide bond would be shown like this. So in the first case, peptide bond is shown between 2 atoms. And when we show also these atoms, we show groups that involved in peptide bond. So both answers would be correct. It just depends on which level we consider peptide bond. On the atomic level, it's going to be carbon and nitrogen, and on the level of the groups, it's going to be carboxylic group and nitrogen group. The correct answer to question C is going to be B. And what, for example, D show us? It shows us R group of the amino acid. Each amino acid has the same backbone and unique R groups. As you see, it is very different for each amino acid. And for example, in glutamic acid, we can find here secondary carboxylic acid. So this is going to be primary, which we can find in the backbone, and this is going to be secondary. And for example, this E, this arrow shows alpha carbon. This is carbon which connected to 4 different groups, 4 different atoms. So carboxylic group here, hydrogen here, and nitrogen or amino group here, and R group. So we call this alpha carbon. And for example, in the case of this amino acid and this amino acid, these carbons are going to be chiral. And as for the alpha carbon that is in this glycine, it's not to be chiral, it's going to be achiral, because it connected only to 3 unique groups: carboxylic group, hydrogen group, and amino group. And here is another hydrogen, so it is achiral, because it connected only to 3 unique groups and not 4. Because central alpha carbon in glycine is going to be achiral, we say that glycine is going to be optically inactive, and these 2 amino acids are going to be optically active. Actually, all the rest 19 amino acids going to be optically active except glycine. And this is all for today. Thank you for your attention and see you in the next video. Goodbye.